Okay, hello everyone and welcome to the masterclass for today. My name is Emily and I will be going through a couple tips and tricks as well as teaching you guys a lesson. Today is going to be on enzymes and I'll give you some good background on the AP exam and some really good studying tips as well. So we're going to start off by talking about here at Master Your Courses, we have a 3-pillar approach that I think is really important for everybody to get to know because it's going to be really important for studying for the exam. Now, the main thing that I find with students is they really struggle with the vast amount of content, and that's completely understandable. I've had trouble with it myself. In terms of this, these 3 steps will kind of make it easy for you, and it should be your 3 steps to helping you achieve a 5. So the first one that we always like to stress is foundation. Now, something I see with a lot of students is if they are struggling with practice problems, they tend to just keep going with the practice problems, and that's not really helpful. What you need to do is kind of go back to the foundation, to go back to the beginning and really understand those foundational concepts like the ones that we learn in Unit 1 and 2. Now, I really stress Unit 1 and 2 quite a bit. You're looking at the chemical context of the cell, so you're getting a good chemical background, and you're also getting the parts of the cell as well, okay? Because when we get into cellular respiration and photosynthesis, which a lot of students come to me and say is really difficult, if you don't understand those foundations that you built in Unit 2, where you're learning about the actual cell and everything that's coming in and out and why, maintaining regulation and homeostasis is so important, then you're really going to struggle. And for biology, everything sort of builds off of one another. So if you don't understand Unit 2, you're not going to understand Unit 3. And you might have already begun to see that, but understanding those foundational units is going to be really important. And it's also going to help you out if you do come across a question that, you're not super familiar with, or a question that maybe you haven't seen before. If you have good foundational knowledge, you can really use that and then apply it to a lot of concepts, okay? So for example, having a deep understanding of how proteins work is really helpful for the enzymes unit, because all enzymes are proteins. If you really understand the foundations of how proteins are made and how they're influenced, you can really understand enzymes a lot better. Okay, so number 1 is foundation. Number 2 is the right type of support. So what we like to do here is provide you with a lot of support in terms of getting all your questions answered and then also having it be very individualized to you. So usually it takes a lot of time to look through textbooks, look online. Not every source is going to give you the same answer for your question. What we have are experts that are going to be able to give you as much support as you need, give you accurate and good support, as well as just help you with whatever you need, get your questions answered, give you extra questions, and really start to understand you. So it's also very individualized support. So we specialize in very individualized support. I understand that, students learn a lot differently, especially with large content-based things like biology. I personally am more of a visual learner. I like to talk about it and I like to visualize it rather than kind of just sitting down and studying notes. But it is dependent on everybody, and we like to support in whatever way you like to learn. Now, the final one is practice. Arguably the most important one, practice, practice, practice. Now, I really stress this because when you're dealing with large amounts of content, sometimes memorization is not the way to go. Understanding is, however, when you need to do all these questions, you want to be able to practice them so that it gets a little bit more ingrained into your brain. You maybe forget about a certain concept and you think, oh, I did a practice question, about this, and this is what I said. So then you can use that and, apply it to something else. So practice is really going to help you have it ingrained into your brain, as well as you're going to get used to a lot of the different questions. And for the AP exam, especially with those application questions, this is really, really helpful because the application questions are about 50% of the exam. And you're looking at questions that you've never seen before. And so there are strategies that you have to build, things like being able to read graphs, understanding the breakdown of these questions, what they're specifically asking for, being able to be clear and concise. Those things are going to come with practice of those application questions. So we will do lots of practice, and our tutors provide you with more than enough practice. We always have more worksheets ready for you if you want to do more practice, and we always recommend doing as much as you can before a test. Now, here are some testimonials from our past students who have used this method, just showing you guys that this method and these tips are really, really helpful. and sort of the style that we do our teaching. I'm going to get on to our lesson in just a couple minutes, but just this style of teaching has been really helpful. So one of my students, Shreya, came to me with about looking at around 70s. In bio, we managed to bump her up by 94% up until her first test. So that was really great. We had another student who got bumped from a 77 to a 94 in just 3 weeks. And then finally, we recommend you guys can watch these on our Facebook page. But we had Shivam, who was entering our program at around 60%, and he ended up earning A's in both courses and actually finished with a 4 on both of his final calculus and chemistry exams. Okay, so this is a really good method and we recommend it to all of you. And as I go through the lesson plan, you're going to be able to sort of see how I incorporate those, and you'll get some study tips as well. So there's just a little bit of background on me. My name is Emily, and I'm currently one of the AP Biology tutors at Masterclass. If you join our Grade Boost program, you'd likely be learning from me. So if you like my teaching style and my approach, you're welcome to reach out. So I just got my bachelor's in biotechnology from Queen's University. Queen's University is quite a prominent school up here in Canada, where I'm from, and I'm currently headed down to the States for my master's in biotechnology at Johns Hopkins, which I'm really excited for. I've had 5 years of teaching experience, so I've only ever had teaching jobs. I love to teach. And then I've also actually completed AP Biology myself, and I've done quite well in it. So I think it's a unique perspective. I understand, what students struggle with. I struggled with it myself, and I know it's not an easy course. I find, teachers will teach it because they understand it, and they won't teach it from the perspective of students that are definitely struggling, and it's their first time digesting this material. So I like to lay it out as simple, as concise, and as straightforward as possible for students, and I can kind of come at it from my perspective of what's the easiest way I would break this down. And it really helps with students, and it also helps connect with students. I always love giving advice and stuff like that. So I've been through it myself. I do know it's very difficult, and I've done enough studying for this exam that I've got a pretty good idea of what they're going to be asking you for. I've also had lots of internships, working for biotechnology companies and doing a lot of cancer research, which is what I really love to do. And now I'm currently working for this company, and I'm helping people get a 5 on their AP Biology exam. So I'm really enjoying that right now. So this is going to be our last slide before I get into the lesson. And so this is going to be my tips to study for biology. Obviously, it's not going to work for everybody, but this is what I've found, especially with dealing with such a large amount of content. I mean, at this point, I've been studying biology for about 6 years, so I've kind of gotten into the groove of the best way to kind of retain all this information. But if this doesn't work for you, then no worries. But these are just some tips that personally work for me. So the first thing I always like to do is to write out notes to increase memorization. Now, reading it on a computer screen is a lot more difficult to have that retain in your head rather than being able to write out and really take time into what you're writing. So I love highlighters, different colored pens, sticky notes, all those. If that helps you with memorization, then that's great. Even if maybe you're not, it's a lot of content, so even just writing flashcards, even for like the more difficult definitions and things like that, or drawing diagrams, just having some sort of component of being able to draw and write things out is going to be really important for you. As well, even if you just like to type your notes on the computer, I also recommend just printing those out and having a hard copy. You can draw on them, you can write notes on them, highlight. I just really love having something physical there. It helps with memorization. Looking at diagrams and visuals. I'm a very visual learner. I know a lot of students are, and I find it very, very helpful for biology, especially when you're thinking of, abstract concepts that you can't really understand, like the processes that are happening in the cell, right? You're never going to see that happen. So having, even just cartoon visuals is quite beneficial in that understanding. So if I don't understand a concept, I like to, you can just pop it into Google and look up images. Usually you'll get some sort of cartoon visual or some sort of diagram, and that really helps you place everything together. The next thing is to take lots of breaks and not to cram. I would not recommend cramming for biology. Coming from experience, memorization takes time. The whole point of memorization is you got to do it consistently over a large amount of time. You can't just try and cram it all in a couple hours. So definitely leads us into our next one, which is review a bit every day. So again, I wouldn't recommend waiting until the day before your biology test. I would just say, read over what you did in class every single day, and you'll be able to, one, retain it a lot faster, and then you won't be as stressed before the test, because stress and a large amount of content is not going to help with memorization at all. The next one is a really good one that I always stress, is that you start with basic multiple choice questions and then going on to long answer when studying. And that goes down to our foundational knowledge that we talked about. Now, basic multiple choice in terms of, definitions, just getting the general knowledge and just making sure you understand the concepts. So doing basic multiple choice is really good. And then going on to the long answer, those FRQ questions, the EP biology FRQ style questions, once you have a understanding down. Because if you don't have your understanding down and you try and do those questions, you're going to get very overwhelmed. They're not going to make any sense. So always do multiple choice questions first and then go into the long answer questions. Okay? The next one is another really important one, is that the best way to test is to explain concepts out loud. And this is what I love to do in my classes with my students, especially for those Q&A sessions. And if I'm getting a student prepped for a test, we usually just sit down, and instead of, just answering multiple choice questions one after another, I like to sit down with them and we have an active discussion about the concepts, and explain this to me. And obviously I'll help, steer you in a direction, kind of help you along with the question, as well as sort of probing if you're not getting as specific as you should be. And that is really the best test to see if you know something, because you might be able to answer all the multiple choice questions, but if you get to the test and it's a long answer question and you're not able to, fully write it down, you're going to really struggle. So saying it out loud means that you can also be able to write it down, and that's the true test of being able to understand and then be able to show that you know what you're talking about. And then finally, understanding, not just memorizing. Again, that goes back to our foundational knowledge and just making sure that you're not just memorizing the concepts, because again, you're going to get questions that you've never seen before. And if you've never seen the question before and all you did was memorize how to do the question, then you're not going to end up being able to get the question. But if you have previous understanding and a good deep knowledge of it, you'll be able to answer, any question, right? So those are my tips for biology. Now we're going to get on to enzymes, and the enzymes unit, so unit 3 includes enzyme, cellular respiration, and photosynthesis. So we'll likely do cellular respiration after this. And the enzymes unit is not one that students particularly find difficult. It's relatively short, and if you have a good understanding of unit 1, then it makes this a lot easier, okay? But some students do struggle with it because there is a little bit of aspect of chemistry. You're dealing with reactions, you're dealing with the idea of energy. Students that take chemistry, tend to have an easier time with this, especially if they've done like reaction kinetics. But this is for AP biology students. So even if you haven't done chemistry, I'm going to explain it really in depth. So even if you haven't taken chemistry, you should still be able to understand it. Again, also still being able to pull knowledge from unit 1, which gave us, our basis for chemistry. Now for the units, and for enzymes, I've broken it down, and this is how I like to break them down into big sort of chunks. Because if you just kind of are dealing with, 6 pages of notes, it's quite a bit. But if you can organize it like this into kind of themes and things you should know for every theme, then it makes it a lot easier to digest. And this is actually the entire chapter for enzymes. So it's not really that much. I usually would break it up into thermodynamics, which we'll start with. So that's the basis. You're looking at the laws of thermodynamics, catabolic, anabolic, spontaneous, and non-spontaneous. The next one you should be able to do is activation energy diagram. So you should be able to label an activation energy diagram, explain which one's exergonic, endergonic, Gibbs free energy, transition states, and all that. The next one I would say is enzyme specificity, so understanding of active sites as well as the importance of structure of proteins. And then finally, likely I wouldn't say the biggest ones, but the most tested would be factors affecting enzyme activity. So you need to know temperature, pH, enzyme concentration, and substrate concentration. That's it. There's only those 4 that you need to know. And again, that still pulls from unit 1 because you're dealing with proteins, so it's whatever affects a protein would affect an enzyme. And then another widely used one for testing is enzyme inhibition, because it'll come back when we start looking at more complex processes. So you're looking at competitive, non-competitive, and allosteric regulation. So these are the big ones. The most tested ones in terms of long answer, I would say, were the ones that fall under enzyme inhibition and factors affecting enzyme activity. Activation energy diagrams are likely multiple choice questions. Okay, they'll likely just, maybe give you a graph or some sort of statistic on a graph, and then they will ask you to explain from the activation energy diagram, is it exergonic or endergonic? Enzyme inhibition and factors and sometimes specificity are more so going to be long answer questions, and thermodynamics is just like a background in it. It's just kind of the intro to it, but you still need to know it. But it's unlikely you'll get questioned on it. Okay, so I'm just going to jump right into the lesson. I'm going to try and get through all of it, but still kind of keep it in depth. If anybody has any questions, you are welcome to text them in the chat. I can see them if they go into the chat, and I can respond to them. Okay. So we're going to start off explaining metabolic pathways. This entire unit is about metabolic pathways. Now, the totality of an organism's chemical reaction is known as its metabolism. We're going to look at cellular respiration and fermentation and photosynthesis and all those methods of metabolism. And in a metabolic pathway, a specific molecule is altered in a series of defined steps, resulting in one product, each step catalyzed by a specific enzyme. Okay, so you're going to be having the synthesizing or the breakdown of a very specific molecule. In cellular respiration, that molecule would be glucose. It's being broken down by an enzyme at every step, and it's being converted into energy in the form of ATP, for example. So that would be a metabolic pathway. Now, our metabolic pathways can be broken into two types of reactions. Now, these are big general reactions. If you do chemistry, there's very specific reactions, but these are big general ones that go kind of by two rules. So there's catabolic pathways or catabolic reactions. Now, they release energy, which is the first key thing here. So release energy. I also have this little tablet here, which I like to use to draw and circle things because you can't see my cursor, so I'm just going to be circling and drawing things. So release energy by breaking down complex molecules. So release energy and then breaking down. So an example is cellular respiration, what I just talked about. You're breaking down a molecule of glucose. And then the anabolic pathway is going to be the opposite. So if catabolic pathways release energy, anabolic pathways consume energy. And if catabolic pathways break complexes, they actually build complexes in anabolic pathways. So these are the two main differences between catabolic and anabolic, and your pathways in metabolism are going to fall into either one of these. So an anabolic pathway would be photosynthesis. You're making a larger complex of glucose, right? So catabolic is releasing energy and anabolic is consuming energy. That's the best way to look at it. If we look at this diagram here, we can see that this is the best way, again, to visualize. So even though this isn't an actual reaction, but it's the best way to visualize it in the sense that anabolic reactions, you have these 4 separate subunits. You add energy onto the reactant side because energy is going into the equation, and then your output is going to be 4 of them linked together. So you can imagine that as maybe even just polypeptides being linked together. That is an anabolic reaction that would require input of energy, okay? And where this energy lies in relation to the arrow is very important. So if it's on this side, this is a reactant, and we're gonna see catabolic, it's a product, because in catabolic, it's releasing energy. So energy is escaping, it's leaving. So catabolic reaction would be the opposite. So for example, you have a big molecule and then you're breaking it down into 4 subunits, and that releases energy. So your energy is a product in this case, it's leaving, and anabolic, you see that it is entering as a reactant. Now, these are the same equations, one is going forwards and one is going backwards. And that is how anabolic and catabolic reactions work. It'll be anabolic one way and then catabolic the other, right? So this one, you can make it going anabolic, and then you could break it going catabolic on the other way. And so the energy going in and the energy going out, you always make sure to look at where the energy is, and that's going to be our big difference between them. Now, this brings us to energy coupling, and this is a very important concept, and you've already touched on energy coupling. You just haven't realized it yet, but I will explain it and it'll make a lot more sense in terms of ATP and why we've been talking about, ATP. So energy released from downhill reactions of catabolic pathways can be stored and used to drive uphill reactions of anabolic pathways. Okay, so that's a mouthful. However, if you think about it, so in catabolic pathways, energy is being released, right? If energy is being released and you need energy to run an anabolic reaction, what you can do is you can put a catabolic reaction first and an anabolic reaction after it, and then you can use the energy generated in the catabolic reaction to drive the anabolic reaction. So this one doesn't show energy as a reactant. So this one might be better to show you guys, because that one's more of a real life example. But let's erase this. Okay, so you would start off with a catabolic reaction. So you would break down something and you'd produce energy. Now you have this large volume of energy, and you can then use that energy to give it to an anabolic reaction. So obviously these would be separate reactions, but if you can see, energy is a product of catabolic reaction and it's a reactant for anabolic reactions. So if you start with a catabolic one, you can produce energy, and then you can put an anabolic reaction immediately after that that'll take that energy and use it to get your final product. Okay? As long as this is more than enough energy to drive this process, you can couple catabolic and anabolic reactions together. And because these molecules will possess energy due to the arrangement of their bonds, breaking those bonds or making those bonds are going to result in the release or the absorption of energy, like we saw. Now, biochemical pathways enable cells to release chemical energy from food molecules and uses energy to power life processes. So catabolic reactions being like the breakdown of glucose. And you can use anabolic reactions after that, such as using it to drive some sort of process. Now, a catabolic reaction that we've talked about a lot that you probably haven't realized is ATP. Now, if you think about the structure of ATP, if you haven't been told, it's adenosine triphosphate. It's an adenosine, we'll just abbreviate it as an A, with 3 phosphate groups attached. What you do when you want energy is you take off that final phosphate right there. So you're doing a catabolic reaction, you're breaking a bond, and that releases energy, right? So if you've noticed, and I like to think about active transport as the most basic example, and you went over it in Unit 2, is if you break down a compound like ATP, you're releasing energy, and then you can use that to drive a process like active transport. And we saw to do active transport, you need to break down an ATP. And that's why, because ATP is catabolic, it's going to release energy. Then the active transport uses that energy to drive its process. Okay. So in this case, you can see that glucose and ATP is being broken down, and that energy that is made from, or the energy that is released from the breakdown of ATP was used to drive the synthesis, so an anabolic reaction, of glucose and fructose, thus making sucrose. So this is catabolic because we're breaking down this ATP, and we can use it to drive an anabolic process, such as putting these two together to make sucrose. Okay, so that is a coupled reaction. Now I'm going to get into the laws of thermodynamics. So for thermodynamics, you're looking at any sort of change or transfer of energy. Now there's 2 laws. There's 3 laws that you go into, but there's only 2 that are important for this curriculum. You're not going to really need to know about the third one. In chemistry, yes, but for biology, the only important ones are the first 2. And the first law states that energy of the universe is constant. Energy can be transferred and transformed, but it cannot be created or destroyed. So that's very important. The amount of energy in the universe is never going to change. Whether you turn something from a liquid to a gas, a solid to a liquid, or you take molecules and you put them together, you take polypeptides and you put them together, or break them down, the amount of energy in the universe is never going to change. You're not creating energy and you're not destroying energy. The amount of energy before the transformation is the same amount of energy after the transformation. You might see a decrease in energy, but that energy that is lost is going somewhere. Okay, so for example, breaking down glucose down to the amount of ATP, you obviously have a loss in energy, but a lot of that energy is lost as heat. That's still energy, it's heat energy. And that's released into the universe. So if you were to add up the amount of heat energy and then the energy of your products, it's going to equal the amount of energy in your reactants. So the amount of energy before and after is always the same. You cannot physically create or destroy any energy. The energy of the universe is constant. Okay? So during every transfer of energy, some energy is converted to thermal energy and released as heat. So again, if you do see a decrease in energy, like we're going to see with, our exothermic reactions, that energy is released and either captured by something or, it could be released as heat energy. Still energy and still the same amount of energy. However, it's just in a different form. Now, the consequence of the loss of usable energy as heat to the surrounding is that energy transfers are going to make the universe more disordered. Now, I'm going to explain what that means. So, scientists use the word entropy, and entropy is something that students struggle with, but I like to think of entropy as just synonymous with like disorder or randomness, because it is a matter of disorder or randomness. And again, entropy and these like chemical or physical concepts are quite difficult to understand. That's why I like visuals. So these visuals definitely will help you kind of envision what I mean by, increasing randomness or increasing disorderedness. So the more random arranged a collection of matter is, the greater its entropy. So what it's saying is, if you have a transfer of energy, you're converting it from one form to another, or you're rearranging bonds, and you have a loss of energy. That energy, obviously, because we know it stays constant in the universe, is lost from the products, but it's released into the universe. Every time that energy is released into the universe, you're making the universe more disordered. You're adding more random particles to the universe, thus increasing its entropy. Okay? So if we look at this one here has low entropy. Okay? It's very ordered, or it's not random at all. I like to think of this as very ordered. It's in a perfect grid. They're perfectly beside each other. There's little room for them to move, and they're very ordered. If you increase in entropy, you're increasing in randomness, you're increasing in disorder. Now all these particles are not together at the same distance. There's some over here. You don't have any sort of order to them. They're a lot more disordered. This would have a high entropy. Same thing with down here. This one's highly ordered. Everything is fitting together. They're very close to one another. You're increasing in entropy. They're going to spread out and they're going to be less ordered or more disordered. So this has a greater entropy. Okay? So you can just think of it as the randomness of the particles. You increase the randomness, you're increasing the entropy. Okay? So every time a reaction happens and it releases energy into the universe is getting more disordered. So this is the second law of thermodynamics. And it says that every energy transfer or transformation increases the entropy of the universe. Again, because for an energy transfer, you're going to lose some energy as heat. That heat energy goes into the universe, makes it more disordered. Now, we're going to introduce you to two concepts and two definitions. And I'm going to connect them all at the end. So I'm going to explain them separately, and then I will connect everything at the end. So we're going to talk about spontaneous versus non-spontaneous reactions. Now, if a given process leads to the increase in entropy without the input of energy, the process is called spontaneous. Now, this is very important. And it is the increase in entropy, but it does not need an input of energy. Now, this might not be the proper scientific way to think about it, but I just like to think of spontaneous reactions as reactions that happen on their own, right? And I mean, you're not inputting any energy. It doesn't need any help to drive the process. It's just going to naturally happen, and it's going to happen on its own. You don't need to put energy into it, and it's going to increase entropy. So making something more disordered or more random is not going to require any energy, is what that's saying. And we'll use this diagram here because it'll be the most helpful. Now, a process that leads to a decrease in entropy and requires energy is non-spontaneous. Okay, so this is a process that won't happen on its own. You need to put energy into it. So non-spontaneous. I like to think of it as a hill, and we're going to see the hill when we talk about activation energy. But you can think of a spontaneous reaction being, you have a ball at the top of the hill and you just need the ball to roll down. You don't need to input any energy in there. The ball's just going to roll down the hill on its own. Non-spontaneous would be something like pushing the ball up the hill. You need to put energy into that. It's not going to go on its own. So spontaneous would be downhill. Non-spontaneous would be something that's uphill. if we look at this diagram here, this diagram I really like, and it puts it into a good amount of context in terms of spontaneous as well as entropy. So if we start on the left side, and this is saying that to go from the left to the right, you're increasing in spontaneity. This is a spontaneous reaction, okay? So we have all of our particles on this left little part here, okay? So they're pretty ordered, okay? They're not disordered. And then by increasing and going in spontaneous direction, so again, this happens on their own with no input of energy, you're having the particles spread out to both sides of the flask in this case. If you look at the left versus the right, you're increasing in entropy, you're increasing in randomness because your particles are farther apart. They're not all packed nicely together in this one little side. Now they're all spread out and they're moving. And this also would not require any input of energy. And the way I like to think about this is in terms of diffusion, right? We all know that diffusion from last unit does not require any input of energy. What particles naturally want to do and what solutes naturally want to do is spread out, right? You pour sugar into water, the sugar is not just going to stay as a lump on top, it's going to try and spread out as far as it can. Particles naturally want to diffuse. So if particles naturally want to diffuse, it makes sense that you wouldn't have to put any energy into it to make these particles spread farther apart. That's what they naturally want to do. So that is a spontaneous reaction. It happens on its own because particles naturally spread as far apart as possible. Now, going the opposite direction would be non-spontaneous. So going from the right to the left is non-spontaneous. You are decreasing entropy. You're taking these really disordered particles that are spread apart and you're putting them into one side. You're increasing their order and you're making them less disordered. Okay, so you're putting them into one spot and that's going to require the input of energy. You've decreased the entropy. You've decreased randomness. That's going to require an input of energy. The way you can think about it is with that same sugar, if you have all the sugar molecules, spread out throughout the water, and then you try and, if you somehow wanted to be able to bring them all back together and pull all the sugar out, that's going to take energy. The sugar molecules will naturally spread out, but they won't naturally come together. You would have to put some sort of energy or do something to allow that sugar to come back together. So to bring things together and decrease entropy is going to take energy. So it's a non-spontaneous. Okay? So this brings us to Gibbs free energy. Now, I'm going to explain this concept and we're going to look at these diagrams and you're going to get very used to these diagrams. And these are the activation energy diagrams. And by the end of this, you should be able to label every part of this, okay? It's a very common multiple choice question. So Gibbs free energy, which is also just known as free energy, is the portion of energy in a system that can perform work when temperature and pressure are uniform throughout the system. So you're just looking at energy change given that everything else has stayed the same, Obviously, adding pressure and temperature and everything and changing all of those aspects are going to affect energy, but we're saying that everything is kept the same. It's a closed system. We're just looking at energy. Now, delta G, so whenever you see a triangle with something, it's a delta, which is the Greek letter, but that usually just means change in. So it usually means you're subtracting two things from each other, so change in. So delta G would be the change in free energy, represents the difference between the free energy of the final state and the free energy of the initial state, okay? So the final state being products and the initial state being reactants. Now our delta G is on our y-axis here. So this is showing us how much energy the reactants and the products have. We're only focused on reactants and products. We're not going to focus on this hill right here. I'll discuss that in the next couple slides. So reactants are starting off at a high Gibbs free energy, and products are at a low Gibbs free energy. So your reactants are starting out high, and then it's dropping down in energy to become products. So your delta G in this case would be the difference, again, between your final state and your initial state. So the distance between your reactants and your products. So that distance from here to here is your delta G. Your change in energy between your reactants and the products. It's not including all of this. That's not what we're looking at. We're going to explain that. However, if it's just between your final state and your initial state, you're just looking at reactants over here at that delta G and products at this delta G. That difference is going to be your change in energy. Okay. Now, depending on change in energy, it's going to determine whether your reaction is exergonic or endergonic. Okay, so an exergonic reaction was the one that we looked at here. This is an exergonic reaction. Exergonic reactions have a very simple rule, and it states that the reactants will be at a higher energy level than the products. If this is our free energy, again, this is our Gibbs energy, your reactants are starting out here, and then it's dropping down and going to a lower energy level in the products. This is what ours did up here. Our reactants are higher than our products. Now, because you are dropping down in energy, that energy has to go somewhere, and that energy is released, okay? So because you're going from reactants to products, you're dropping an energy level, that difference in energy is released. So this releases energy, and this is an exergonic reaction. So an exergonic reaction proceeds with a net release of free energy. The delta G is negative, and they occur spontaneously and is energetically favorable. An example is cellular respiration. So the way I like to remember this, and a really good trick that all my students use, and I still use to this day because exergonic and endergonic sound very similar, is that for exergonic reactions, the EX, I think X as exiting. So in an exergonic reaction, energy is exiting. Okay, so that's how you always remember that energy is going to leave for an exergonic reaction. So if you look at this, like, oh, the reactants are higher than the products, energy is leaving, energy is exiting, that's an exergonic reaction. And the delta G is also going to be negative, okay? So this is also really important because the change from products to reactants, so the change from the final state to the initial state would be negative. The amount of energy, the delta G, is going from here to here, which you can see here, and that's negative. This is a negative because you're decreasing. You're decreasing in energy, that's going to give you a negative change. So the delta G for an exergonic reaction is always negative. And then also, if we think back to our spontaneous and non-spontaneous reactions, spontaneous reactions require no input of energy. They actually release energy. So this is a spontaneous reaction because you're having no input of energy. You're actually releasing energy. So when I said that I would explain all of them separately and then come together, it's that exergonic is always spontaneous. You're never going to have an exergonic non-spontaneous reaction. That's physically impossible. So although I explained both of them and we know what both of them mean, they're pretty synonymous with one another. Exergonic is always spontaneous. So exergonic equals spontaneous, and it also equals a negative delta G. It's never going to be a positive delta G. So all three of these go together. This also goes together with catabolic reactions because catabolic reactions release energy, remember? So catabolic reactions are always exergonic, they're always spontaneous, and they would always have a negative delta G. So all of these go together. You're never going to have them mismatched, okay? So if you are asked, I've seen a question, they gave you a negative delta G actually. They said on the AP exam, glucose breaking down, it's negative, 100 and something kilojoules in change in free energy. So negative, and then they asked you, is it exergonic and spontaneous? Is it endergonic? Is it non-spontaneous? And the answer for that one, if it's a negative delta G, the answer was exergonic and spontaneous. That was all the question was. So exergonic, spontaneous, negative delta G, and a catabolic reaction. So the magnitude of this delta G for an exergonic reaction represents the max amount of work that the reaction can perform. The greater the decrease, the greater amount of work that needs to be done. Okay, so this means that when we're looking at the release of energy, you can have different magnitudes of delta G. The amount of delta G is going to indicate how much energy was released. If you had, negative 100 versus negative 120, negative 120 is saying that you're releasing more energy. Okay, so the magnitude is also very important. The negative versus the positive is going to indicate whether it's exergonic or endergonic, but there is also the idea of the magnitude of the delta G. If it's negative 120, it's releasing 120 kilojoules of energy. If it's negative 100, it's only releasing 100 kilojoules of energy. Okay? And then an endergonic reaction is going to be the exact opposite. So your reactants are starting off at a lower energy level than your products. So to get from reactants to products, if you're going up in energy, you have to absorb energy. Okay, you can't just go up on your own. You need to absorb the energy and the exact energy difference between reactants and products. Okay, and that change in energy is going to be your delta G, obviously, and it's going to be positive because you're increasing in energy. You're going to have a positive delta G. You're absorbing energy to get up to your products. So an endergonic reaction absorbs free energy, the delta G is positive, and the reaction is non-spontaneous. If we do remember, non-spontaneous reactions can't happen on their own. This reactant, if it's starting at a lower energy, cannot turn into a product on its own. It needs energy. Non-spontaneous reactions always need energy. So endergonic, so remember exergonic exit, endergonic equals non-spontaneous. So this is not going to happen on your own. You need lots of energy. It's going to be a positive delta G because you have to put energy into it. And these tend to be anabolic reactions, right? The building of something, which is going to require energy. Okay? So that's endergonic versus exergonic. And you definitely need to be able to determine the difference between them and then remember, which one's spontaneous. But if you just write these out, it kind of takes 4 definitions and kind of just puts it into one. Okay, and they're always going to go hand in hand. So even if you do have a multiple choice question like the one that I mentioned where, they said, is this exergonic non-spontaneous, exergonic spontaneous, endergonic spontaneous, or endergonic non-spontaneous? Obviously, 2 of them don't go together or never going to happen. So that whittles it down to 2. And then you just have to decide whether, energy is going in or energy is going out. So that is exergonic and endergonic, and keeping, putting it all together. Like I said, being able to, make flowcharts or, make concept maps is really important because you can put a lot of things together and make it easier on yourself to memorize. So I've just cut down, a couple minutes of studying by just connecting those concepts. Now this is what the activation energy graphs would look like. Again, reactants is starting up high and products is starting down low. So that's a negative delta G in terms of energy being released. And then reactants starting low for endergonic and then going up high to products. That's going to be a positive release of delta G. Again, it's always, we're not including this hill. I'm going to explain what this hill part is on the next slide, but it's just between its reactants and products, just that distance. You're not including the hill. Okay. You need to be able to look at this graph. So if I gave you the shape of this graph and, I obviously didn't give you this top bit about endergonic and non-spontaneous, you have to be able to tell me whether it's exergonic or endergonic. Obviously, you look at where reactants are and where products are. You could also maybe tell me, is this a positive delta G or negative delta G? You should be able to tell me if this is spontaneous or non-spontaneous just based off the blank graph. Okay. So if you aren't able to, go back, rewatch what I just said, and then you should be able to answer all those questions just based off a graph. And that's a common multiple choice question that you will see. So now I'm going to explain this hill that I've been alluding to. And this is actually very important, but in terms of free energy release and exergonic versus endergonic, it does not have an effect. What this does do is it indicates that when you want to reach a state where the bonds can change the reactant, it must absorb energy from their surroundings. When the new bond of the product molecules form, the energy is released as heat. Okay, so the initial investment of energy to start the reaction is going to be known as the activation energy. And that activation energy, it's abbreviated as EA like this, is the distance from reactants to the top of that hill. So our EA is here, what I'm doing here, that's our EA. Our delta G still is always just reactants to products. It's this yellow line here. So if you obviously added up the delta G and the EA, that would take you to the top, okay? But EA is just reactants to the top of the hill. That top of the hill is called the transition state, and I'll explain that in the next one of what that means. However, what this is indicating, and this is an exergonic reaction, but the same thing goes for an endergonic reaction, is that for the reactants to be converted to products, there has to be an initial absorption of energy. Now, I know I said that exergonic reactions have a release of energy, and they do. They have an overall release of energy. But every reaction needs to absorb a little amount of energy to get converted to products. And that's because, in this, you can see that the bonds are being rearranged, and most chemical reactions are going to involve the rearranging of bonds. Now, in this one, A and B are linked together. in our reactants, and then our products, we have A and C linked together. So you have a bond breaking and a bond reforming. Those don't just happen spontaneously. They're not going to happen randomly. Actually, they're not spontaneous processes. They're non-spontaneous processes. You have to put a little bit of energy in to basically weaken those bonds and make them unstable and ready to break. They're not just going to break randomly. But if you put a bit of energy into them and you weaken them and you get them ready to break, it's going to make the process go a lot faster. Now, you need to have an initial investment of energy, and that's that EA. It's going to absorb, so your reactants are going to absorb a little bit of energy from the surroundings, usually in the form of heat. So if you put heat into something, you're obviously going to speed it up. But if you have your reactants absorbing a little bit of heat from their surroundings, you're going to have those bonds weaken, you're going to get those bonds ready and very unstable, and then they're able to break and then recombine immediately after. But you have to have an initial investment of energy. And that EA is what's being absorbed from the surroundings to make those bonds unstable and ready to break. So activation energy is the amount of energy needed to push the reactants to the top of an energy barrier, and it's often supplied by heat in the form of thermal energy. So the reactants have to get to the top of their activation energy barrier. This is not an option. If they only absorb, let's say, energy up until this point, where I'm putting in red here, the reaction is not going to happen. You're not going to get any products. You have to get to the top of that barrier. It's called a barrier because you kind of have to jump over it, is what the reactants have to do, is get over that top part. Okay, so if you absorb enough energy to get to the top, to that transition state, then the reaction will happen. If you don't, then the reaction is not going to happen. If you get halfway up, it's not going to work. So it's kind of like, again, the example of the ball going up the hill. The ball's not going to go down the hill unless you push it to the top of the hill. Okay, you got to get the ball to the top of the hill before it rolls down the hill on its own. Okay, if you push the ball halfway up the hill, it's just going to fall back down. It's not going to go over the top of the hill and then go to the bottom. Okay, so you have to put an initial investment energy. You have to get your ball to the top of the hill before you push it down. Okay, now I'm going to explain what the transition state is, which is that highest point of our activation energy, this point here, that's alluded to right here. So the transition state, you need to be able to label where the transition state is and as well as explain it. So when molecules have absorbed enough energy for the bonds to break, the reactions are in an unstable condition known as their transition state. And this is the most unstable state of the molecule. Okay, so they might ask you, I've seen it as a multiple choice question, when is it most unstable? Always the transition state. The transition state is like a split second right before it gets converted to products. Now, your reactants are absorbing energy from their surroundings, their bonds are getting weak, the bonds are bending, they're getting ready, and they're super unstable and ready to break. Once they reach that top where they've absorbed just enough energy to have the reaction go, that's its transition state. You're going in between reactants and products, so it's in its transition. This lasts for a split seconds when the bonds are most unstable and ready to break, and then once those bonds break, they rearrange, and then you go to the products. So transition state is the most unstable, so this is incredibly unstable because it has a very high energy. When you have a lot of energy in something, it's usually unstable and easy to break. So incredibly unstable, it also has the highest energy, but you have to reach that transition state where the bonds are basically ready to break before you get to products. Now, this activation energy barrier provides a barrier that determines the rate of reaction, since the reactants must absorb enough energy to reach the top of the activation energy barrier. Now, this is very important, and that's going to bring us into one of the most important things that we're going to cover is how enzymes do this. But this is a barrier. You need to jump over it to get to the other side. Now, the higher the barrier is, the longer it's going to take you to get over it. Okay? So the higher the barrier is and the more energy that you have to absorb to get to your products, that's going to affect the rate of reaction. Okay, so how the rate of reaction is how fast you're making your products, how fast are you converting your reactants to products. If your reactants have to absorb a lot of energy to be converted to products, it's going to happen slow. Okay, so the higher the activation energy, the slower the rate of reaction. And this brings us to enzymes. And if you're going to take away anything from this class, it should be this, because you need to know the mechanism of how enzymes affect reaction rate. Okay? So enzymes are macromolecules that act as catalysts that speed up reactions without being consumed. Okay? So they're macromolecules. They're specifically proteins. Every single enzyme is a protein, okay? It's a protein. So they will behave like proteins. They are made like proteins. They're made of amino acids. They fold the exact same way from what we've learned in Unit 1. They are proteins. That's why we learn Unit 1 first, and we learn about the background of protein. So now when we talk about enzymes, you have a good idea of, what's important about proteins, what's important about their shape and all of that. And a catalyst is anything that speeds up a reaction. Now, I just want to mention, because you learn about catalysts in chemistry, a catalyst is anything that speeds up a reaction, but an enzyme is specifically a biological catalyst. It is a catalyst that's made by the cell. Okay? Some catalysts in chemistry include like magnesium and those, but those are not enzymes because they need to be made by the cell. So the cell will make the protein, and I'm just basically saying that every enzyme is a catalyst, but not every catalyst is an enzyme. Right? Enzymes are just catalysts that are made by the cell. In biology, you're only going to be dealing with enzymes. Okay? And they speed up a reaction without being consumed. So they are not a reaction and they are not a product. I'll show you a bit about what I mean, but they go in and they come out the same way. They don't get their bonds rearranged. They don't get affected by the reaction anyway. They just kind of come in and then they help. So without regulation by enzymes, chemical traffic through pathways of metabolism would become terribly congested because many chemical reactions take too long of a time. Now, what's important to know from this is that reactions can still occur without enzymes. All these reactions naturally occur. They just don't occur very fast, okay? Or they might not occur fast enough to, sustain like an entire body or something like that. So the reactions will still happen. They just happen too slowly, okay? And sometimes it's just not enough to keep up with your body. So you need enzymes to help speed up those processes, like speed up your metabolism, speed up digestion, and all those things. But they do still happen on their own as long as you reach that activation energy. So heat can also increase the rate of reaction by allowing reactants to attain their transition state more often. Again, if your reactants need to absorb heat from their surroundings to get to the top of the activation energy barrier, if a reaction or if your reactants that are in, a solution or something are heated up, you're gonna have more heat energy to absorb. There's more energy available for you to reach the top of your activation energy barrier. So heat increases rate of reaction in this case, but we're gonna talk about that more in detail when I get to like the factors that affect it. So an enzyme catalyzes a reaction by lowering the activation energy, enabling the reactant molecules to absorb enough energy to reach the transition state. And that's probably the most important thing that you're gonna get out of this masterclass. This part here is the most important. You need to know the mechanism by which enzymes speed up chemical reactions, okay? And that is by lowering activation energy, and so that the reactants then have to absorb less energy to get over their activation energy barrier. So if we look at this diagram here, we have two lines. We have one that is without an enzyme and one that is with an enzyme. Now this black line here, if we look at its EA, its activation energy barrier, so from its reactants, so the amount of energy the reactants have to absorb to get to their transition state, it is this much energy, okay? Up to the black line. Our red line here was treated with enzymes, so it had an enzyme. You can now see that the activation energy is a lot lower. The amount of energy that the reactants need to absorb is about half of what it needs to absorb regularly. So look at that difference in the amount of energy that has to be absorbed. That's how reactions are sped up by enzymes. And that's the most important part. You will 100% get a question on your test and on the AP exam about that. Enzymes lower the activation energy, and that speeds up the reaction because your reactants then have to absorb a lot less energy. They basically get to the top of the hill a lot faster, and then the reaction can happen a lot faster, right? If your activation energy is half of what it's used to, you're going to be doubling the rate at which those reactions are going to occur, okay? So it lowers activation energy. But the one big thing that you need to also keep in mind is that it will never change the delta G. So if you look at where our black and our red lines, which is the same exact reaction, one is just with an enzyme, one is without, their reactants and their products start at the exact same energy level. So they are both still starting with reactants at this energy level and products ending at that energy level. So the delta G is never affected by the enzyme, and that's very important. I've seen multiple choice questions where they will ask what the mechanism is, and one of the options is always, that they change the delta G. That will never happen. The enzymes never change the delta G. They're not going to change how much energy the reactants have, how much energy the products have. They only change the activation energy. So how much the reactants have to absorb to have that reaction go. Okay, so it's never going to change the delta G. If it's spontaneous, it will always be spontaneous. You're not going to change anything like that. Your delta G is never going to be affected. The only thing that's going to change is your activation energy. And the activation energy is always going to be smaller for reactions catalyzed by an enzyme. Okay. Now, the last things I'm going to talk about is substrate specificity. And so when we talk about enzymes, there is something very important. And like I said, I'm a visual learner. And the best way that I like to think about this is through the lock and key model. And it's a popular way to envision it, but it's the best way that I like to think about it. So the reactant that the reactant acts on is the substrate. So we have an enzyme, and what's coming into the enzyme is called its substrate. And when the enzyme and the substrate come together, there's a very creative name for it. It's called the enzyme substrate complex. So whenever you see two of them come together and you have to label it, it's just the enzyme substrate complex. It's when both of them come together. Now, the reaction catalyzed by each enzyme is very specific. An enzyme can recognize its specific substrate even among closely related compounds. So only a restricted region of the enzyme molecule binds a substrate, and this is known as its active site. Now, this is where the lock and key model comes from. If we look at this enzyme here, its active site, the active site is just the little, you can imagine it as a little pocket in the enzyme where the substrate comes in and binds. It's the binding spot for the substrate. The enzyme will have an active site with a very, very specific shape. Now, if we remember, enzymes are proteins. We know that proteins fold into very specific shapes. This is why. We mentioned enzymes and that's why protein folding is important, but this is specifically why, and it's because of the active site. Enzymes will fold to have a very specific active site shape. And this is very important because your substrate has a matching or complementary shape to that active site. And they're going to fit together like a lock and a key. And they have to fit together for the reaction to occur. If your key doesn't fit in your lock, you're not going to open the door, right? So they have to fit perfectly together for the reaction to happen. If they didn't, then you're not going to have a reaction. You're not going to open the door. So the active site has a very specific pocket that binds a very specific substrate. So these two only go together. A lot of the times it's a 1-for-1. One enzyme is only responsible to bind one specific substrate. They make this lock and key model here, and then you get the reaction. If they don't come together, or if one's the wrong shape, or a substrate that's not meant to go to this enzyme comes in contact with this enzyme, there's going to be no reaction, right? So the specificity of the enzyme is attributed to its complementary fit between the shape of its active site and the shape of its substrate. So again, specificity is because an active site has a very specific fit with its substrate, okay? The lock has a specific key, and the key has a specific lock. This also helps with fine-tune regulation. Now, what I mean by that is, let's say your body is producing too much of a certain protein or a certain hormone, okay? You're producing a lot of hormone and you shouldn't be producing anymore. Your body is basically like, we have too much of this now, we need to stop producing it. You're telling your body to stop producing more of this hormone because you already have too much. If you can just turn off the enzyme or degrade the enzyme, and that enzyme is specific to that hormone, turning off the enzyme turns off the hormone, right? So if I were to draw this, you have an enzyme like this, and it has like a little triangle pocket shape, and it only has one substrate. If we have too much of the substrate, we would just stop producing the enzyme, and then you would stop that, right? It's a 1-for-1. However, if you were to have one enzyme that catalyzed a bunch of different reactions, so we're not thinking about complementary fit here, if they were able to do, let's say 3 substrates, and we produce too much of our triangle 1. We produce too much of this hormone. If you shut off the enzyme, you're shutting off all these other ones too. Sometimes you only want to turn off one. So if you have a 1-for-1, it's easy to control it and have a very fine-tuned response. But if you have one enzyme that acts on a bunch of different substrates, it's very hard to regulate each one of those substrates individually. You can't. So they're usually very, very specific to allow your body to specifically regulate the concentrations of everything in your body, okay? Now, most metabolic reactions are reversible, and an enzyme can catalyze either a forward reaction or a reverse reaction depending on the direction of the negative delta G. By now, you should already know that a negative delta G means something that is exergonic. So, enzymes will catalyze exergonic spontaneous reactions. Now, reactions involving 2 or more reactants, the active site provides a template on which the substrates can come together in proper orientation for a reaction to occur between them. Now, sometimes it's not just one substrate coming in. Sometimes if the enzyme's job is to bind 2 things together, you're going to have 2 substrates come in. The enzyme is going to make that bond between them, and they're going to leave as a pair. In this case, we have the reverse happening where they're leaving separately, but it would have 2 active sites or an active site that fit, having both of them beside each other, and that helps bring them together. It also speeds it up by having a little pocket where both of them can come together and fit, so you can easily make that bond. Now, the active site might also stretch the substrate molecule towards the transition state form. So this is also how enzymes speed up the reaction. Okay, this is the mechanism. Either way, it speeds up how fast they get to the transition state. And this is sometimes called the induced fit model, but let's say that it's not always the most perfect fit, but the enzyme can kind of take the substrate and make it fit the active site. That's why it's in the induced fit model. So sometimes the enzyme will change shape slightly as the substrate binds. So again, there needs to be complementarity though. They're not just going to bind anything and, make it shape fit. It's going to bind something that fits specifically, and then it'll kind of be able to manipulate the substrates coming in and stretch the bonds and get them more unstable. So by stretching the bonds, making them more unstable, you're moving them closer to their transition state, right? Because transition state is when the bonds are super unstable and they're ready to break. So an enzyme might also help to sort of make those bonds unstable and weak, and that's going to, increase your rate of reaction. They also might provide a microenvironment that's more conducive to a particular type of reaction. So we've seen that a good environment for a reaction is like having 2 of your substrates that need to be bound together, coming together in the active site. Or maybe for the reaction to happen, it has to be at a specific pH, or you need specific temperature, things like that. It's providing an environment that is best to have that reaction occur. It's going to manipulate the bonds, it's going to manipulate the activation energy, and it's going to give it basically the best opportunity to be converted into products so that it happens as fast as possible. In this case, this enzyme is breaking apart 2 substrates. So you have, this one's coming in as a whole. Again, it's still complementary fit, so they're going to come in here. They're connected to one another. You're going to have the enzyme substrate complex, and then you're going to break them apart, and your products are them being separated, and then they're going to leave. So this would be a decomposition reaction. But if you can see, the enzyme in the gray from here, and then once it releases the products, is exactly the same. It does not change. The bonds are not broken. It does not get reincorporated with products. That's why we say enzymes are reusable, and they are reusable, because the way that they go in is the same way that they go out. Right? You're never going to have your enzyme change shape, break bonds, do anything in terms of the actual enzyme or protein itself. So enzymes are reusable, and I've also seen that as an AP exam question. Why do we say enzymes are reusable? Because they go in the same way that they go out. If they come in that shape and they leave in the same shape, they can then go and these get reused, because this will just go on to the next reaction. The next step for this one is to go back to the start and have, the substrates come in and bind again, break apart, substrates come in, break apart. So enzymes can constantly be reused, and that is why they're considered reusable. And that's also why they're not considered as part of the reaction, because they're just kind of external helpers. They're not actually being rearranged, or, they're not being integrated into the products or the reactants. They're just kind of a helper that comes in and then leaves. Now, I'm going to finish this off by talking about this last little question here. We have a couple more things to go, so I might do that on the next masterclass. However, I did want to finish with, like a little question that's here. And this question is really good because I like to ask my students this because, like I said, building connections is very important. You're going to have questions on the AP exam that's going to put together a bunch of different units and maybe connections that you haven't thought about or haven't seen before. If you start doing it now and you think of it all as an interconnected sort of continuum, then you will have a lot easier time making those connections. Now, like I said, we learned about macromolecules in Unit 1. And so we're going to be connecting Unit 1 and then enzymes right here in Unit 3 to answer this question. And I have seen this as an AP exam question. It was obviously differently formatted. This is just sort of a bigger open-ended question, but something along these lines has been on the AP exam where you have to connect, primary structure and enzyme function. Okay, so this question says, using everything you've learned from Unit 1 and 3, or 1, 2, 3, explain how the primary structure of an enzyme determines its function and specificity. And I've added a little hint here that says, why is shape so important to the enzyme's functionality? Now, a lot of my students, I taught this today, and they said, well, we understand, secondary and tertiary structure and how that would affect function and specificity, but we don't understand primary structure. You got to take it back all the way to Unit 1 and understand those fundamentals. So what is primary structure? Okay. Primary structure is just the sequence of amino acids. The sequence of amino acids. So for example, if you have, we'll just write out their one-letter code, so like lysine, arginine, we can just add a bunch of random ones here. Okay. I think that should be good. So this is the primary structure. There's no folding involved at all. So you're thinking, if shape is so important to the enzyme's functionality and specificity, then what would primary structure have to do with it if it's just the unfolded amino acids? However, if you do remember how the proteins fold, it is because of the R groups and the actual amino acid sequence that is determining the folding. So the order of the amino acids is very important for how it folds because it's going to determine which ones are interacting with one another. Let's say in the tertiary structure, you have the R groups. This might not be super accurate, but we're just going to guess that L and T interact with one another. Can't remember off the top of my head their R groups, but let's just say that L and T interact. Okay, that is going to cause a fold. Okay. And then maybe M and R interact. That's going to cause another fold. So what the sequence of amino acids is going to determine how it folds because, L is always going to react with T and M is always going to bond with R. Let's say you, for some reason, you don't have a T here. Something happens and now you have we'll have an S here. Now L doesn't ever bond with S, so there's going to be no bond there. You're going to get a different shape because there's going to be no bond. There's usually a bond here, but now there's no bond. So the sequence of amino acids is ultimately determining the secondary structure, the tertiary structure, and the quaternary structure. Primary structure never changes. That's why a protein can always refold if it's denatures and you put it back into its regular environment. It can always refold back to the same way it was if the amino acid sequence stays the same. The amino acid sequence is never going to change unless you actually break apart the protein. But if it just loses its shape, it's going to refold because the same amino acids are going to interact with one another. Okay, so the sequence of amino acids determines the folding. Every protein has a different sequence of amino acids, and they're going to have different folding. Okay, so the primary structure determines its function and specificity. So primary structure is going to determine, because each one of these has an R group, they're going to make hydrogen bonds, hydrophobic, hydrophilic interactions. That determines secondary, tertiary, and quaternary structure. Now that we've established that, how would then primary structure affect the function and specificity of the enzyme? Well, when we're folding it into its final form, what else is being formed? The active site. So you also, when you fold it needs to fold properly so that you have to fold a proper active site as well. Okay, so fold into proper active site. Because of that specificity and because of that lock and key model, if you didn't have this bond here, for example, you didn't have that, maybe your active site would not be the correct shape, right? You would have a missing bond in your active site. Now when your enzyme and your substrate come into contact, if your substrate is going into an active site, that's misshapen, they're not going to have a reaction. So that's going to determine its function. An enzyme that isn't folded with a proper active site is not going to be functional. And it's also going to affect its specificity, right? If you need to have a specific shape of your active site, having improper folding and an improper shape is ultimately going to affect your active site. So it needs to fold into proper active site, and that is going to be dependent on the amino acid sequence, because amino acid sequence determines folding. R groups, the R groups are very important for tertiary structure specifically. And if it does not fold properly, you're not going to get a proper shape of active site, then you're not going to get any function because your substrate is not going to recognize it, and you're not going to ultimately get a reaction. Now, an example that I believe we went over in Unit 1, at least with our Grade Boost program, and one that they love to ask questions on is about sickle cell anemia. Now, the concept of sickle cell anemia I kind of used already, but sickle cell anemia, you have, let's say these two again interact regularly, and then these ones interact. Sickle cell anemia is a mutation where it changes one amino acid, just one amino acid. I think it's like a 200 amino acid sequence long, but just one of them is changed and it's swapped out. It's a mutation. We don't know for what reason, just one of them is switched. That affects the folding of the protein, right? Because now maybe the regular amino acid makes a bond usually, and if you swap that out for another one and now there's no bond, or it makes a different bond somewhere else, you're changing the shape of it. You've changed the shape of that, and it actually extremely affects the functionality because the sickle cell makes red blood cells. And so when you have a sickle-shaped red blood cell, it goes from being shaped like this. Changing one amino acid causes it to be shaped like a crescent moon. That's why it's called sickle cell. This now doesn't have the active site for O2, so this usually binds O2. This cannot bind O2 because it's the wrong shape. And people that have sickle cell anemia are obviously anemic, and they don't get enough oxygen through their blood. So you can cause very detrimental diseases, even just one change in the amino acid. So one change to that primary structure is going to fully affect its function and specificity. Sickle cell anemia is a disease. That is an example of that, but you need to have proper folding, proper active site because of that lock and key model, and that all goes back to the primary structure of the protein. Okay, so we are probably going to finish this lecture off on the next one, but I will skip forward. So we just had factors affecting enzymes and then enzyme inhibition. And then I wanted to go over a 2022 AP exam question. This is one from the enzyme unit. So this one's actually not too difficult, but it's a really recent one. And then we'll go over the answer guide as well on the next one. But I just wanted to give you guys a brief little background on our program since you guys stayed for the class, and hopefully you enjoyed it. I put a lot of effort into these slides and teaching these classes. So if you do like that, I'm also the person that teaches the Grade Boost program here at Master Your Courses, and I'll just give you a little bit of a background on that if you guys are interested. Now, our whole thing is that we want you guys to get a 5. And if you see, I'm very focused and the curriculum is very focused on the AP exam. I know what they ask most likely on the AP exam, what they don't ask, the way they ask the questions, and my goal is to get you guys a 5 on the AP exam, and I will try and do everything I can and give you all the resources that I can to do that. And that's going to be through our Grade Boost program. And so a little bit of a background on what you can kind of expect if you get involved in the Grade Boost program or just with our company, is that you can expect, live classes with our experts. So if you enjoyed this one, I teach the Grade Boost programs, and we have a lot of great tutors that are, at the top of their classes. And you can expect live classes. So if you did enjoy this, you can expect a lot of those. Q&A sessions to tackle application questions. Now, we specialize in doing lots of Q&A sessions because, again, one of our pillars is practice. Now, we want you guys to practice those application questions because it's 50% of the AP exam. It's 50% of your mark, and teachers will not do a lot of practice with those, and you need to practice. You need to develop strategies. You need to be able to understand those application questions because it's 50% of the exam. It's basically the make or break. The multiple choice questions, if you study hard enough, most of them you can get, but application questions are where we specialize in helping students, okay? So those Q&A sessions, we just go over difficult questions. I have all the past AP exams, all the questions organized by chapter, like I already showed you one from 2022, as well as all the answer guides. When we go do the one with the answer guide next, you'll be able to see how important the answer guide is for understanding how to do those questions. So I always, it's not always good to look at the answer first, but sometimes it is best to see, what they're looking for. The specificity, how in depth they want it, and you're going to get a good idea of what they're looking for. The next is direct communication for homework help and questions. Now, I directly communicate with all of my students. On our platform, we have like a chat function, and they reach out to me. I've had students reach out to me regarding, homework questions. They're studying for a test, and they just ask me, I don't understand why this answer is the answer. I'll shoot them a text back and, help explain it. If they, ask for extra practice problems or help before a test or anything like that, or they want me to re-explain something, they mention that to me and they just directly message me. So in terms of the right type of support, you have someone on your side basically 24 hours. And I personally work full-time for the company, so you're having someone that can message you throughout all the work hours and answer all your questions. So, the night before a test, or if you have a project or an assignment coming up, you have a tutor that will reach out to you and give you all the help you need, so you don't have to look on the internet or go through your textbook or really waste a lot of time. You have someone that understands you and understands the curriculum. We also have practice questions for every unit. I don't know if you guys saw on our Facebook, one of the last posts was about our AP biology book, and that's something that I spent a bunch of months on to make as organized, as simple, and straightforward as possible. And it's going to be your entire guide to understanding what you need to know for the AP exam. It has outlines of all of the units. And so my personal, notes that I've taken, everything is well organized. I have things highlighted, lots of diagrams, the way that I like to, if you're a visual learner, there's tons of visuals in there. And then at the end of every unit, there's actually, 30 to 40 practice questions, as well as some application questions. So reach out on Facebook if you don't already have that, because it is a good tool if you guys are studying, and it's good to have, your kind of just pure guide for the AP exam, and it's at the depth and the level that you should know for the AP exam. Some teachers don't teach it at the proper depth, and then they expect you to just do the application questions on your own or practice mock exams on your own, but this will be, if you learn it at this depth and you know everything from the book, you are bound to get a great mark on the exam. We also have AP mock exams and tests. So I always give, practice tests, practice problems before my students have tests. We also do that in the Q&A session. And then our book also has a full mock AP exam at the end, completely structured like a AP exam. So the 60 multiple choice, 6 long answer FRQ questions with an answer guide. Our book has, the answer for every single question. There's no really point in doing questions if you don't know if you're getting the answer right. So there is a full answer guide in there. And then finally, individualized learning plan for every student. And this is kind of what we're most proud of, and what we focus on, and how we get the best results is because it's very individualized for every student. Like I've already said, I know that students learn quite differently from one another. Some are more visual, some struggle with concepts, some struggle with memorization, some struggle with application questions. It really depends on the student. So what we do is we will take you in for a consult if you're looking to join our program. And again, if you did like this and you liked my teaching style and all the resources that we have, I believe Masood has put the link to book in a meeting with us in the chat. So that's just a consultation meeting, and it's completely free. And you can chat with us if you're interested in the program, and you'll be talking to me as well as Masood. And I'll come from like the biology side, and we'll talk about what you're struggling with, and we'll kind of make a plan for you. So it's very individualized. We sit down with just you, and we figure out, oh, I'm struggling with either this particular unit, or I'm struggling with application problems. If you're struggling with application problems, then maybe we do, a couple Q&A sessions a week, and just practice those before your test or something like that. Or maybe you're struggling with concepts, we can do more lecture-based ones. So it's very individualized for every student, and so you're welcome to book in a consult with us, and we'll kind of help you out. Even if you don't end up doing the program, we can give you, materials or tips or anything that you need, because we really want you guys to do well on the final exam. It is, I won't lie, a pretty difficult exam, but if you follow all these tips and you get a little extra help from us, you guys should be doing great. So the link for that is in the chat. I can see it right now. You can click that and book in a meeting with us at any time, at any of those available times, and we will do a consult with you, and we can give you more information on the program as well in terms of how many classes and pricing and all that. But again, it is very dependent on the student, so we have a wide range of things. It really does depend on what we say in the consultation in terms of like if you're needing more lecture-based, more Q&A based, anything like that. So that's something we'll figure out. So you're welcome to book in a meeting with us. We'd love to chat with you. And yeah, that's it for my masterclass. For the next masterclass, it'll still happen next Saturday as well. I'll probably just do a last little bit of enzymes, and then I'll move on to cellular respiration, which will definitely be good, and I would definitely say tune into, because cellular respiration is one that I get requests for all the time. It is one of the hardest units. I struggled with it when I was in school, so I wouldn't be too stressed if it is something that you're struggling with. I have a good way of organizing it, very simplified, and I know the depth that you need to know for the exam. So I'd say tune into that. We're going to go over cellular respiration, which should be super helpful. And yeah, I will stick around for the next little bit if you guys have any questions in the chat. If not, I will see you guys either in a meeting or on the next one.